Bueno, buenas tardes a todos. Vamos a empezar. Bueno, pues el día de hoy tenemos con nosotros a la doctora Sylvie Girard, que viene de la Universidad de Montreal y ella trabaja en el Centro Hospitalario de Saint-Justine y trabaja en el Departamento de Obstetricia y Ginecología. Y a pesar de todo eso, no es médico. Es bióloga y es inmunóloga. Entonces, es muy interesante hablar con ella justamente porque hace esta interfase entre la medicina con el paciente y la investigación, y que ahora es lo que llamamos la medicina translacional, ¿no? Entonces, ella trabaja justamente en la relación madre-feto, en la interfase madre-feto, y su trabajo está enfocado a la inmunología, pero justamente ayer con los estudiantes tuvimos una excelente clase de cómo es que se pueden abordar estos problemas que pueden parecer tan inmediatos desde puntos de vista mucho más amplios, como es justamente incorporando conocimientos mucho más grandes, como son todos los que nos ofrecen las ciencias ómicas. Entonces, también quisiera decir que con nosotros están nuestros aliados de siempre, que son el Centro de Biotecnología Genómica de Reynosa, también en Mérida nuestros colegas de la UNAM, y aquellos que están siguiéndonos de manera individual, pues muchas gracias por estar con nosotros. Les recordamos que el chat en vivo está en la liga www.lcg.unam.mx/frontiers/live. Y bueno, doctora Girard, muchas gracias por estar con nosotros y fue realmente un placer. Gracias.
Muchas gracias. Thank you. I hope this was nice. Sorry, I don't really speak Spanish, so this will be in English. Thank you so much for inviting me and for welcoming me. I had a great time yesterday with the students for the Genomics program. We had a great chat. And so yesterday I spoke a bit more about omics. Today there will be a bit of omics, but I'll especially talk to you about my work, which is focused on perinatal inflammation. And so the relationship between the mom, the placenta, and obviously the baby. So first of all, just before getting into the work, so I just want to introduce to you pregnancy complication. So there's three of the most important pregnancy complications that are known. We have IUGR, intrauterine growth restriction, when the baby is smaller than it should be. You have preeclampsia, which is characterized by maternal hypertension, and I'll talk a bit more about this later on. And then you have preterm birth, when the birth is happening too early. Now, what's interesting is that in all of these pregnancy complications, inflammation has been detected. So this has been really the common factor in all of this pregnancy complication. Now, all of them has been associated with a higher risk of mortality as well as morbidity in affected women as well as in the babies. And for example, in the newborns, we call it fetal programming, where the baby is programmed to develop, for example, neurodevelopmental disorder, such as cerebral palsy as well as schizophrenia. But these babies and the mom as well are also at higher risk of cardiovascular pathologies later on in life. And so just before talking to you more about what I do in my lab in terms of inflammation, I just really wanted to clarify infection versus inflammation. So we all hear quite a lot about infection. So we know that infection is caused by pathogens. We use E. coli in the lab quite a lot, which is as a classical inflammation inducer, which is derived from pathogens. Now, what we know is that infection are always leading to inflammation. So this is quite well known. The thing is, most of these pathogens will be blocked by the placental barrier. So in most pregnancy complications, you will detect inflammation without any infection. So now one of the questions that we ask is even if there is no infection, how come there is still inflammation? And this is why we've been studying quite a bit what we call sterile inflammation. So I don't like to use the word sterile because I think nothing is sterile, but this is what we use when we say endogenous inflammation. So inflammation is very important during pregnancy because there is really tight regulation of the immune system, which has to be done to allow proper pregnancy to success. And then any imbalance in inflammation will have an impact both on the mom, on the placenta, and obviously on the fetus. Now, from now on, when I will be using the word inflammation, so we know that inflammation is needed in life, especially even in pregnancy. But when I'm using the word inflammation, now I'm talking about non-controlled inflammation, so much bigger inflammation than there should be, and it's negative action. So the link between inflammation in the perinatal period and the impact on the child has been quite well studied. And there is quite a strong association between perinatal inflammation and the risk, a higher risk in the babies of having especially brain injury. So now what we ask is this due to endogenous mediators of inflammation that could be involved? So we've been focusing on the DAMs, so damage-associated molecular patterns, also known as alarmins, which are basically molecules that you all have in all of your cells in your body. And these molecules will usually be within the cells. So when they are out of the cell in higher concentration than they should be, they can cause inflammation. And they have basically the capacity to activate the same receptor as pathogen. So basically the PRR, so the TLR, for example, the tool-like receptor, which are the most widely known receptors for PAMs and DAMs. So here I just put a few examples of the alarmin that we've been studying in the lab. Some of them have been studied as well in the context of pregnancy. Uric acid, HMGB1, which I'll speak a bit more today. You also have the S100 protein, cell-free fetal DNA, and quite a lot of others. So think about basically anything that you have in your body that you need. When it's in too high concentration, like IL-1 alpha, for example, then it can lead to abnormal inflammation. Now, uric acid, I'll speak a bit more about it today because we've done quite a few studies on uric acid. And why? Because this molecule is elevated in the maternal circulation in women with preeclampsia. So we've been studying this, obviously in women with preeclampsia, but we've also seen elevated uric acid in women without hypertension. So there is a lot of people that think that if you have higher uric acid in preeclampsia, it's because the maternal hypertension is having an impact on the kidneys. But we know that in pregnancy pathologies without hypertension, such as, for example, high UGR, you can still have elevated uric acid. Now, just before moving into the actual work that we did, I just wanted to talk briefly about the cause versus consequence question. Every time I give a talk, I'm being asked, is inflammation the cause of pregnancy complication or is it a consequence? And actually, I think it's a bit of both. So I'm not going to give you the big answer today about if it's a cause or not. I think it's a bit of both, depending on when, depending on the situation. Inflammation can cause a pregnancy complication. It's been shown to lead directly to preterm birth, for example. But I think in other cases, like in preeclampsia, it can be a consequence. But even if it's a consequence, there is inflammation there, and it really highly contributes to the inflammatory cascade, which is being seen within the placenta and in the mom. And this has quite a lot of bad effect on both the placenta and on the baby, especially through the irreversible damage that are being done within the placenta. So then what we ask, so we made the hypothesis, we have the maternal immune cells. I want to put here. So you have the maternal fetal interface here. So these are cells within the placenta. And within these cells, so you have endogenous mediators of inflammation. So when there is stress happening within the placenta, cellular stress, you're going to have necrosis. And these are going to be released. So for example, here I've put uric acid, HMGB1. These are just examples. There is quite a few others as well. And these are being released within the maternal circulation and will affect either the trophoblast cells, which are cells from the placenta, which are going to be the neighboring cells. And you also have the maternal immune cells within the circulation, which are going to be affected. And then these will then lead on to having an impact, a systemic impact on the mom circulation. So then the way for us to study that was obviously using in pregnancy, where we have three interrelated compartments that we have to work with. So the first one that we focus on is the placenta, which you can see here. And so basically the placenta is the only way there is a direct contact with the maternal circulation. So you have the maternal on one, the mom blood on one side, which is in contact with the placenta. And so the placenta can release debris and damps within the maternal circulation, and it can also release it on the other side. So you have the placenta, hang on, here. The placenta which will release damps within the maternal circulation, and then it can have an impact not only on the mom during pregnancy, but also after. And then on the other hand, You also have the release from the placenta of inflammatory mediators, but in the other side, and then having a strong negative impact on the baby's development. So then based on that, what we set out to do, so our objective was really to understand what does perinatal inflammation do on the placenta and how does it impact on both maternal health and fetal health, and especially through the placenta. So I'll be speaking a bit about how can we use the placenta as a way to maybe predict early damage within the babies. And on the other hand, how can we also study inflammation and how can it lead to an impact on maternal health? So these will be the two parts of my talk. So the first question we ask is how can we use the placenta to maybe identify early on high-risk babies? So this had lead to two questions. So can those placental changes be linked to abnormal neonatal development? And if so, we can then identify those babies. So the way we set out to do this is that we recruited women within the St. Justin Hospital, and we got their placenta within 30 minutes after our deliveries. And then we did an analysis of the transcriptome, so using RNA sequencing. And we also did histological analysis. Why did we do both? Actually, histological analysis is used quite widely. For some reason, it's not used that much by the clinicians, the obstetrician, even though it's done pretty much routinely on all of the patients. And I think a lot of information can be taken out of these analyses. So we've set out to do both this more visual analysis of the placenta and then combine with the in-depth RNA sequencing analysis. So now in this part, we set out to study all three pregnancy pathologies. So I've told you that the common factor to all three pathologies was inflammation. So then we wanted to see, is it the same inflammation? Is it different kind of inflammation that's happening? So preeclampsia, I've told you already a bit about this. So it's characterized by hypertension happening in the mom after 20 weeks of gestation. And then you have two kinds of PE. So you have the early onset PE, which will happen between 20 weeks to 34 weeks of gestation, which is the more severe phenotype of preeclampsia. And then you have the late onset, which is the less severe phenotype. So we've split them both because even though these are the same, they have the same clinical characteristic, I mean, one of the phenotypes, like the early PE, are much more severe, so they are quite distinct in terms of their presentation. We also had women with preterm birth, so delivery before 37 weeks of gestation. And now here, this is preterm birth without preeclampsia. A lot of the women with preeclampsia end up delivering early, but in this case, we really wanted to see the impact of gestational age without preeclampsia. And then we had the group with IUGR, so intrauterine growth restriction. So smaller babies, but at term. Because we do have smaller babies that are born preterm, but then you have the impact of gestational age. So we really wanted to minimize the confounding factor within these different clinical variability. So here I'm just showing you different pathologies, and I'm not going to go into too much detail here. So this is basically the clinical data that we got from each of these patients. And the difference that I just want to show to all of you is that obviously the gestational age is quite different. So the group with early PE, they were born much earlier on, so 32 weeks. Late PE, they're born close to term. Preterm birth, obviously 32 weeks as well. And then IUGR at term. And then you also had the birth weight. So like I told you, babies in the group of women with early PE were much smaller, those with preterm birth as well. And those with IUGR were smaller, although they were not as small as in those two other groups, which had much more pathology, obviously, added pathologies. So now what we set out to do, so we just did an RNA sequencing of all of these placenta. And the first thing that we asked, so we've adjusted for sex. And after that, we just wondered, so are any of these pathologies clustering together? So what you can see here, so basically, so H dot is one patient. And as you can see, so when we do the analysis totally unbiased, each colors are each of the groups. And you can see that pretty much nothing clustered together. The only cluster that we could see here, so we can see that most of the control all clustered here together. We had a few, like one here and one here, that were quite different. But from the rest, most of them clustered together. Now, the next question that we ask is that, so what's common between all of these pathologies? So we know that we can detect inflammation within the placenta of these women. And when I say detect inflammation, is that I mean that we are able to detect any cytokines, for example, within the placenta, or we can detect macrophages or immune cells there. So is there any way, using that unbiased technique, we can also detect inflammation? So we've done a comparison of all of the pathologies, and you can see here pretty much all of the different number of genes that were common to all of these pathology. And the important number here is almost 200, which is the number of genes that were regulated, that were changed in all pathologies similarly. So then the question that we ask is, what are actually these genes? And so, oh, hang on, I'm always going the wrong side. So what are actually these genes? And so what we, here I'm just gonna put, so lots of these genes were regulated, and I'm just gonna put the top pathways for you to see a bit better. And we can see that basically within the top about 7 pathways, most of them were related to inflammation. So immunoregulatory interaction, leukocyte activation, lymphocyte activation, cytokine, interleukin-2. So we can see that basically when we could detect some inflammation in each of these pathologies, but clearly there is inflammatory pathways that are common to all, and potentially these could also be targeted therapeutically, which is something which we're studying at the moment. Now the next question that we ask is what is actually happening within each of these pathologies separately. Like I've told you when I started the presentation, so preeclampsia is mostly hypertension. Preterm birth is the pathology that has been most strongly associated with infection, especially, but also with inflammation, and then IUGR. So we set out to study all of them separately. And so we wanted to see if we could see any inflammatory pathways happening. And so what I'm going to show you here is that both in preeclampsia and in IUGR, for some reason it's not showing up, but you can see that out of the pathways that were the mostly activated, so these related to inflammation, so T cell activation, leukocyte activation as well, and cytokine production. So a lot of inflammation is going on within preeclampsia. And we also saw some inflammatory pathway in IUGR. Lymphocyte activation is the top one here. I'm sorry, it's still so small. And then we have cytokine as well, activation, which is there. For some reason, in the group with preterm birth, this didn't, oh, it's coming here. For some reason, in the group with preterm birth, most of the pathways were all related to the infant development. So we're quite surprised that within the top genes, these were not related to inflammation. So now we're setting out to study, so how come we don't detect inflammation that much? What we think is happening is that in women with preterm birth, most of these are extremely acute events. So women with preeclampsia, usually they come within the hospital, they've been followed up for weeks before they end up having to deliver because the pathology is so bad. So it's more of a chronic kind of inflammation. Whereas in women with preterm birth, they were fine, and then they come in and they are in labor. So it's much more of an acute kind of inflammation. So maybe this acute inflammation that we can detect some cytokine didn't lead to change gene in terms of gene expression within the placenta. But this is something which we're going to set out to study in more detail. Now coming back to the question about how can we use this to identify babies that are going to have abnormal development later on. So we wanted to combine this data with histological analysis. And for those of you that are less familiar with the placental histology, I've just put some example here of what we can see. So these are all placenta that were obtained obviously after delivery. And these are hematoxylin and eosin staining, so H&E staining, which is giving the purple, the fact that the nucleus are colored in purple, and then you have the pink, the eosin staining, which is in pink, and then you can see here the red blood cells, which are seen in pink. So what we did with those placenta, so this is what we have like a normal looking placenta, and then we separated the changes that we saw in two. So we have on one side the structural changes. So you have two examples of what can be seen. So here in the structural changes, you see a lot of those purple nucleus agglomerate. So these are syncytial knots, which are usually observed in women with preeclampsia. And this is what is being shed in the maternal circulation, and it's inducing inflammation. On the other hand, you can have that kind of placenta with a lot of fibrin, which is the pink here that you can see, which is a lot of fibrin deposition, which means that the placenta is not perfused properly and not working well. And then I'm just giving you one example of inflammation. You can clearly see here that the amount of cell is much, much higher in the placenta with a corium neonitis, so very strong inflammation there. And you can see an immune reaction, so a lot of immune cells within the placenta. So for this part of the work, I'm just going to show you, we split the placenta by either normal, structural, or inflammation, which was either mild or severe. So this example is like a severe kind of inflammation. So we've done that on the same placenta that we've done RNA sequencing. And so this is just showing you within each of the groups, so preeclamptic, early, late, preterm birth, and IUGR, how did the placenta work? So you can see in the control, we had one that showed some sign of structural of inflammation. In that case, with early PE, lots of structural damage, late as well, and then some inflammation. In preterm birth, we could clearly see severe inflammation observed in there. And in IUGR, we could see some inflammation as well, even though it was mild. Now, how does that relate with the baby's development? So all of these placenta were obtained within the last year and a half. So now we've done the evaluation until only 12 months of age with the babies. So this is not an in-depth follow-up of the babies. Hopefully we're going to be able to follow them up even more. But I want to show you now what we've been able to do with correlating the placenta with baby's development. And so we've looked at the outcome. So we split them in 4. So either pulmonary problems, cardiovascular disease, neurological disorder, or others. So in the others, we had a few babies that unfortunately passed away. So this is what we put in others. So what we could do here, so you can see that in the control group here, we have pretty much none of the kids with complication. And then what we could see, you can clearly see that in those babies that showed complication, the percentage of patients was related to those that we could see changes within the placenta. So far, this study was done blinded, so to make sure that the pathologists didn't know what they were looking at and that the students didn't know what they were looking at. And right now, we're looking more in depth. Is those patients that we saw, for example, very severe inflammatory pathways, actually the ones that are observed to be sick later on, so we can correlate directly. But then now the exciting part was in terms of genes. So could we use any genes that we detected within the RNA sequencing and correlate those with abnormal babies that had abnormal development? So for this, we went up until 6 months because this was done a bit earlier on. And what we've been able to do so far, so I'm just giving you 2 examples of genes. I'm not saying that these genes are actually causing the baby's abnormal development. I'm just saying that there is an association. So far there is no cause there yet. We're really just studying, and these are just examples. So there was quite a lot that we could see. So I'm just giving you 2 examples. The first one, NTRK2. So this gene is basically the BDNF receptor, so we're quite excited about this one. And we could see, so you have babies with neonatal complication, which are the one with the white bars. And we can see that within the group with preeclampsia, so those were elevated, but these were increased especially in babies with neonatal complication. And then on the bottom side, you can see TBX15. So TBX15 is a transcription factor. And what we could see in that case is that it was elevated in babies from pregnancies with preterm labor, so preterm birth, and even more in those that had abnormal development. So what's interesting in there as well, so I've told you before and I've shown the table that babies from women with preeclampsia were also born preterm. So these differences is not due to gestational age. It's really due to the placenta and the baby abnormal development, and also to the pathology. So the differences that we see are quite sensible enough that we're not just detecting variation in gestational age. I can say that some of the genes were actually indeed related to gestational age, and I'm not showing them here because obviously we know these are related to the age difference. So these we don't want to use them to potentially identify babies at high risk of abnormal development. So now for the first part, just a short conclusion for that part. So we could see inflammatory profile in each of the pregnancy complication. In preterm birth, it's not clear yet, but still this is ongoing. And we could see that the placental gene expression could potentially be used to identify high-risk infant. Now the other part that I want to talk to you about, so I've talked to you about the placenta linking with the baby. And now on the other hand, we're going to talk about the mom a bit. The moms are important as well. So we've been focusing a bit more on preeclampsia because this is a pathology which is extremely having quite a big of an impact on the mom, not just during pregnancy, but also after. So women that had a preeclampsia are much higher risk of having cardiovascular disease later in life, and we don't really know why. So we set out to ask, so is there any inflammatory changes happening in these women with preeclampsia? And is there any changes in the immune cells especially? And could there potentially be a role of these maternal immune cells in the endothelial activation that we see in preeclampsia? So I've told you that the clinical presentation of preeclampsia is hypertension. And so to have hypertension, you need to have endothelial activation. So we wanted to see if these immune cells could potentially be leading to endothelial activation. And if so, what could maybe be the mechanism? So we, again, we recruited women at the St. Justin Hospital. In that case, so what we've done, so we had control women, so normal pregnant women. We had women with preeclampsia. And then we had women with, here set out to be PPPE, which is postpartum preeclampsia. Now I'll just speak a tiny bit about postpartum preeclampsia because I've told you that preeclampsia is during pregnancy, and I'm saying postpartum, so there is no more pregnancy. So basically, preeclampsia can also happen after delivery. So within 48 hours to 6 weeks after a woman gave birth, she can develop preeclampsia. So basically the same clinical symptom, hypertension, another organ being affected, but there is no more placenta there. So what we thought was happening, so in preeclampsia, what people are thinking is that you have, well, you have the placenta. As soon as you remove the placenta, so you give birth, the women's hypertension is going down. So we thought that the placenta was kind of the cause of this hypertension. And we started studying women with postpartum preeclampsia, thinking this is the perfect control because you have a preeclampsia without a placenta. So then we can see what is really the placenta doing in this setting. And now just before we go into the results, a few differences between these women. So you have what we saw the differences. So in most women with postpartum preeclampsia, half of them were from Black descent. And then we had women with preeclampsia, which are mostly Caucasian, which is quite well known. We had also a difference in terms of here the BMI, higher BMI, and also most of them had a history of hypertension or PE. So now here, what is happening in these pregnancies? So what's really interesting about those women with postpartum preeclampsia, these are all normal term deliveries. So you can see here, in terms of the women, so gestational age, they all delivered at term. The babies were the same weight as the control baby. So the differences that we see here are all in women with preeclampsia, similar to the other group I've shown you before. So now what we did is that we took blood before they gave birth, before and after. I'm going to show you most of the results before today. And we basically isolated immune cells within these, from these women. So we did a fecal gradient, and we took the immune cells. So before studying the immune cells, for one thing we did, we took the plasma as well, and we look, is there any DAMs within these women? So uric acid has been associated with PE. So we did saw a strong increase in uric acid in the circulation of these women, which we couldn't see in women with postpartum preeclampsia, clearly showing that even though these are both preeclampsia, these are quite different pathologies. We also studied HMGB1, which is another inflammatory, endogenous inflammatory mediator, which has been increasingly associated with pregnancy complication. And on the other hand, in this case, we could see an increase in women with postpartum preeclampsia, not in a classical presentation of preeclampsia. We've also looked at quite a lot of cytokine, chemokine, and we couldn't detect anything. So now here's this profile, these DAMs, are they associated with any immune profile change within the maternal circulation? So we did quite a lot of flow cytometry on these women, and now I'm just showing you a few of the change that we saw. So we did saw an increase in total lymphocytes within both PE and postpartum preeclampsia in terms of percentage as well as proportions of immune cells. And we saw also this increase in CD4 and CD8 in these women. We did see some slight increase in monocyte, which was not as strong as what we saw with the monocyte. What was really interesting is that we saw a very strong pro-inflammatory bias within these cells, so a Th1, Th2 ratio in the CD4 T cells. Now getting to the exciting part, so can this potentially be involved in endothelial activation? So these women have hypertension. Could these immune cells actually be contributing to hypertension? So what we did is that we took those immune cells from these women, a set amount of immune cells from all women, and we've performed co-cultures with UVAC, which are human umbilical vein endothelial cells, to see if the immune cells taken from these moms could be able to induce endothelial activation. So now I've just shown you that the composition of these immune cells were quite different between like control and women with preeclampsia. So we did use a set amount from all of the pathology, and then we did adjusted for the differences in terms of the proportion of immune cells. So we studied ICAM, VCAM, and E-selectin secretion because these are markers of endothelial activation. And we just went to see in our population of women with early or late PE, are these women actually having higher levels of these markers as well in humans, or are these only markers in vitro? And I can show you that in our women cohort, they have also strong increase in ICAM and VCAM, especially you can see here the levels are extremely high. So we do see endothelial activation quite a lot in the human patients as well. Now, so how did it look in terms of the culture? So we just took the cells from either healthy pregnant, either non-pregnant. So we had the non-pregnant women in there because we see that pregnancy is changing quite a bit, a lot of the stuff. So we have the non-pregnant, the healthy pregnant, and then we have the preeclamptic women. And so we put 300,000 immune cells, 24 hours on top of the endothelial cell layer. And then we looked at, I'm just showing ICAM and E-selectin, and we could see strong increase in endothelial activation, which was induced only by immune cells from women with preeclampsia. Now, are these results due because there is more cells of one specific subtype, so CD8, CD4, for example? I can tell you no, because what we did is we put 10 times more of the cells from healthy pregnant or non-pregnant, and we could still absolutely see no endothelial activation. So it's not because there is more CD4, it's really because of their phenotype. So now the question is what is that actual phenotype? And so this is a question which I cannot fully answer now, but I'll give you a few of the results of what we've seen within this population. So we know that there is an elevated Th1, Th2 ratio within women with preeclampsia. So we looked, is there any increase in the cytokine that these cells are secreting? So we just took the cells, put them in culture for 4 hours, and we look at what they were secreting using multiplex assay. And what you can see here is that as compared to healthy pregnant, the white bar that you can basically not see because there was absolutely nothing there. Those with either early PE or late PE, especially the more severe, so the gray, the pale gray, you can see an increase in pretty much all of the cytokines that were studied, especially IL-6 and IL-1. We've also seen that there was an increase in the percentage of interferon gamma staining. So we did ICS, intracellular cytokine staining. And we can see an increase in the interferon gamma within those CD4 positive cells within women with preeclampsia only. So there is clearly a pro-inflammatory bias going on in these women. So now, is the endothelial activation actually due to the fact that these cells are releasing pro-inflammatory mediators, or is there any cell contact that is needed there? So we performed the exact same experiment, but this time using transwell. So when you have no contact, this is when the cells were put, so the immune cells were put within a transwell. So there could be exchange of like media, but the cells themselves couldn't touch the endothelial cells. And what we could see is that when we did the contact versus no contact, even though you had a bit less of activation without contact, you can still see activation of the endothelium, which led us to believe that the secretion of cytokines of our inflammatory mediators could at least be half or maybe more than half, but partially responsible for the activation that we see in this. So we performed the same experiment, but this time using IL-1 array, because I've shown you that IL-1 was one of the most increased cytokines within these cells. And IL-1 array is the IL-1 receptor antagonist. So we just wanted to see if we added this to the culture system, could we maybe decrease endothelial activation? And indeed, what we could see here, so you have cells from women with early PE, late PE, and then here with IL-1 array. So for the cells with women with late preeclampsia, you could see an almost total decrease. And you can see maybe half of a decrease in terms of the endothelial activation in the cells from women with early PE. So you cannot decrease everything, but you clearly have an effect. So the release of inflammatory cytokine is clearly involved in that case. Now, the other result that I just really want to briefly show you, because I think it's extremely exciting and it shows quite a strong difference between pregnant women versus non-pregnant in terms of their immune function. We use, again, the same setup, so using PBMC, so peripheral blood mononuclear cells. But this time, instead, all of what I've shown you before was just the cells non-treated taken from the women and then put in culture. Now we've taken those cells and we've exposed them to different kind of immune stimuli. So we've used LPS from E. coli as a classical PAM, so a pathogen stimulus. Then we use the uric acid crystal, which is a DAM, so an endogenous mediator of inflammation. And then we use IL-1 because IL-1 is basically induced by both LPS and uric acid. So it's a downstream mediator. So what we see, so you always have the non-pregnant, here you have the early PE, and then you have the late PE. So in the untreated, so UT untreated cells, you can see that there is only cells from women with PE that are inducing, activating the endothelium. With LPS, everything activates the endothelium, which makes sense. Even if we use very low dose of LPS, 10 nanogram, this is still inducing, all of the cells are then able to induce the endothelium, to activate the endothelium. Just telling you here, so we washed the cell very carefully to make sure that there was no crossover of LPS or uric acid directly on the endothelium. And we've also done a control experiment where we directly treated the endothelium with these treatments to see what was happening. And the effect was never as strong as what we see here with the immune cells. Now, where it gets interesting is when we expose them to uric acid. As you can see here, the black bar, so with women, the non-pregnant women, they were not responding to uric acid at all, which makes sense because you would always think what we know from the immune system in adult is that uric acid itself wouldn't be inducing inflammation. You would need another stimulus before to induce inflammation. But in healthy pregnant women, you can see that these cells, they clearly don't need any other stimulus. They could activate the endothelium as strongly as cells from women with preeclampsia. So just a quick reminder, so women with preeclampsia have high level of circulating uric acid. So when you further stimulate them even more, so they don't activate more than they always do, but they have been exposed to uric acid for quite a while in the women. So this means that women, that the immune response is different in women with, that are pregnant. And these, instead of using always LPS, like we all do as a positive control or as a clear stimulus, maybe we should start using some DAMs, because even though in that case, those DAMs don't do anything in non-pregnant women, they clearly have a strong effect on these immune cells from pregnant women. Now, so just to conclude from that part, so I've shown you that there was an altered maternal immune profile in women with preeclampsia, which was associated with placental inflammation. And then we've also seen increased levels of DAM, so uric acid, HMG1 within the placenta. I haven't showed it to you today, but we saw, even though we didn't see any increase in HMGB1 within the circulation, we did see a very strong increase of HMG1 within the placenta. I hope I've convinced you that the maternal immune system was clearly involved in endothelial activation, which is being observed in preeclampsia through the release of inflammatory mediators, and that the immune response is quite different between pregnant and non-pregnant women, and especially not just pregnant, non-pregnant, but also DAMs versus SPAM. So the types of stimuli is also extremely important in that case. So just an overall conclusion of what I've talked to you about today. So I've shown you that we could potentially use the placenta to identify high-risk babies. I think we still have a lot of work to do on that case. How can we use those data we got from the RNA sequencing, combining this data with the histological analysis that we have? Could we maybe just be able to understand better what's happening in this pathology? So I've shown you 4 groups of different pregnancy complications. These are all clearly extremely heterogeneous. So what we see in the clinics versus what we see in the placenta is extremely different. So is there any way we can use these in-depth analysis of the placenta to better characterize what is being seen in the clinic and to help clinicians? And also, obviously, to be able for early identification of baby soon after birth, so then we can treat them early on, or at least follow them more closely. Then on the other hand, I've also shown you the impact of preeclampsia, this time on maternal health, and the contribution of the maternal immune system to the clinical presentation of the pathology. Now, what are we going to do from now? So just a few areas that we're actually pursuing. So I really think that we can use the placenta a bit like an open window on child development. So I think a lot of the organs, especially the brains, were formed during pregnancy, and studying the placenta quite more in depth, we can potentially be able to know what happened to the baby and how the baby development will happen. Now what we're doing is we're focusing on preterm birth, because these babies are much more they're followed much more closely in the clinic, so this is a bit easier for us to do at the moment, but clearly we want to do also in women with preeclampsia or IUGR. Now, in terms of this maternal immune activation happening in preeclampsia, what we're doing is that we're using humanized mice, where we're injecting those cells that we've been using in culture, we're also injecting them in the mouse and see, so are the immune cells themselves enough to induce this phenotype that we see? And on the other hand, we've also looking at epigenetic changes within these cells. So now I've been talking to you about the fact that women had higher risk of cardiovascular disease later in life. So we want to follow these women after their delivery and see if there is any epigenetic memory that is being observed in these cells that could make them more susceptible to later occurring immune stimulus in life. And then the last point, I'm coming back to my 3 interrelated compartments. You have the mom, you have the placenta, and you have the baby. And so we're really trying to make those 3 compartments talk to each other better and to obviously facilitate diagnostic and treatment. And now we're working hard to get obstetrician, pathologist, and neonatologist to talk to each other, which is quite, it's even more complex than getting this molecularly to speak to each other, but it's going to happen. And I just want to finish by thanking you all for your attention. And this is the one that did the work. This is my lab, most of the work that I've presented to you today was done by Marie-Ève and Virginie here. And I just want to thank you all for your attention, and all the women as well that were involved in the work. Thank you.
¿Hay preguntas?
Thank you. Very nice talk. I have 2 questions. One is regarding the defects in the babies that had high levels of the TrkB receptor. Was associated with neurological disorders or more?
We haven't done the analysis yet. It seems to not be just in neurological disorder. So this is why I was very careful saying we see association, no causal. And you have to think these are increased gene expression in the placenta.
Okay.
So we haven't looked like, we don't have any baby like brain samples or anything. So this is really what's happening in the placenta. So I cannot say that what we see in the placenta, we see an increase in this gene in the baby as well. So we were excited because it's BDNF, so we thought maybe these babies are still too young for us to see really any neurological impact. But yeah, we'll see. Hopefully, when we follow them up later on, we'll be able to answer that question.
I assume that this is the full-length receptor, not the truncated form.
I cannot tell you exactly what it was. This was really just the gene that was. We didn't look if there was an impact. You know, if we could. Okay. We really just checked the RNA sequencing at that stage. Yeah, we didn't look at the proteins, for example, within the placenta or anything.
And my other question is related to the second part of the immune response in the mom. So, when you use the basically the whole population of the white cells, right, in the experiments, in vitro experiments, and in those the pump itself is doing the activation of causing the activation of the endothelial cells. So, but you mentioned that in your profile was T cells basically the ones that were changing. So, could you speculate how it will work, how a pump will activate the T cell, or it needs an intermediate cells to have that effect on the endothelial cells?
That's an extremely good question. I think the immune cells are working together. So what I didn't show here, because now what we're trying to see is which one is doing what. So we have really high levels of CD4 and CD8. So we've been trying to split the cells up. So we have CD4, I know we do flow sorting of the cells. CD4, CD8, and then we tried to take the monocyte. We couldn't get enough. So we did CD4, CD8, and the rest. And when we split them, we don't have any endothelial activation. Now, this could be because of not a problem, but the process of sorting the cells, which is making them not being able to activate anymore. Or it could be because the cells need to interact together. So this is all when we don't treat the cells. When we treat them with LPS, for example, I definitely think that we treat the monocytes are reacting, leading to T cell activation afterwards. In that case, totally. I don't think that LPS itself is activating T cells, for example. But I think in women with PE, the cells are already being activated, potentially by uric acid in the mom, in the circulation. But I'm not saying that it's a direct effect on T cells. It could totally be an effect on monocytes and then a collaboration between the immune cells. Yes.
Just a comment. I was in a seminar where they show macrophage with the T cell receptor expression, and those were clearly inflammatory cells. So
okay, maybe we'll have to look at these. Yeah, interesting. Thank you.
Hello, very nice talk. I wonder, have you checked the prostaglandin synthase as an inflammatory molecule? Because we have a lot of it in the activated CD8, neonatal CD8 T cells.
We haven't looked. The reason why is because we didn't look because we knew in women with preterm birth, they probably had higher level because this is quite known to be associated with preterm birth. So we thought we have a pathology which is causing something which would cause this increase. We did look at other stuff like progesterone, for example, and things like that. And we see again a link with preterm birth. We didn't look directly like if immune cells, like in culture, were releasing it. This could be a way we could look at it. So, but this we haven't done. That's a good point.
Yes. And the other question is, well, you have found that it's uric acid may be a culprit. You think perhaps a woman who had previously preeclampsia and is pregnant again, something that dampens the uric acid could help?
That's a very good question. There is not a lot of things that block uric acid. There is a drug that could be used. The problem is not every woman with preeclampsia has elevated uric acid. So It's not like every single woman. Most women have elevated uric acid, not all of them. Okay. So, and women that don't have still have preeclampsia. So this is used in the clinic as a diagnostic marker, but not that much. We've already published that it was inducing quite a lot of inflammation. I don't think it's never been looked at if women that had a preeclampsia before already have higher levels of uric acid. I know they use aspirin to try to decrease inflammation and make sure there's no coagulation. Yeah. But maybe that could be a way, to use that drug to block uric acid.
Yeah. Okay, thank you.
Thank you.
Question.
Is it? Oh, yes. How can we use transcriptomic sequencing results to develop more precise anti-inflammatory therapies for preventing pregnancy complication and adverse fetal outcome? So I'm assuming that person can answer me, can listen to me now. Actually, this is a good point. I don't think we could. Well, maybe we can use RNA sequencing to have more to identify new targets. I think that we already have quite a lot of anti-inflammatory treatments out there. I've talked about IL-1 already. It's already used in the clinic, and it's specific for IL-1. There is quite a lot of drugs which are working in animals. They just don't work in humans. And I think that we don't really know, we're not able to characterize our humans properly. So I think some women could benefit from some of these drugs. So I don't think that having a better drug will help. I think we have lots of drugs. We just don't know who to give it to. So being able to target these women so knowing the pathways and knowing which women could benefit from these, this is the way where we're going to be able to have better therapies for preventing pregnancy complication. And hopefully with these, we can protect the placenta. And I really believe that if you protect the placenta, you're going to protect the baby. But that was a very good question. And there was another one. Do you have any prediction of the genes involved in placenta and neurodevelopment? This is a bit similar to the question I had before, and this is something which we want to do. We don't have enough information yet about the babies to know how is their neurodevelopment going to happen. We're doing another study focusing really on preterm birth, where we're following the babies at 3 months, 6 months, 12 months, and 24 months. So hopefully they have in-depth neurological tests being done on them. So hopefully with these babies, we'll be able to correlate what we see in the placenta and be able to find, I don't think we're going to have one that's going to link with the brain development. I think we're going to have multiple, and then having maybe 10, and then we can say, if you have these 10, then you have a higher risk, and then having maybe being able to have an odd ratio and have a risk factor for those babies. So I think this is the way we could maybe use this. It's a good question.
¿Preguntas?
Awesome. Great. Thank you so much.
Gracias.